next speaker. Our next speaker is Dr. Jianming Yu. Dr. Jianming Yu started his career in plant sciences with a BS degree in agronomy from Northwest Agriculture and Forestry University in Shaanxi, China. He then moved to United States and he got his master's in science in plant breeding and genetics from Kansas State University under Dr. Mitch Steinstra. Then he moved on to University of Minnesota to do a PhD with Dr. Rex Bernardo. He did a year postdoc with Dr. Bernardo and moved on to Cornell University to do a postdoc with Dr. Ed Buckler. From there, he took a professorship position at Kansas State University in 2006, where he was assistant professor from 2006 to 2010. Then he got a associate professorship in Kansas State from 2010 until 2012, when he decided to move on to Iowa State University as associate professor. So now he is a professor and pioneer distinguished chair in maize breeding at Iowa State University. During his scientific career, Dr. Jianming Yu has published over 80 different publications in reputed journals like Nature Genetics, Science, PNAS, the Plant Genome, Genetics, and many more. He has been, over this course, he has been cited over 10,000 times, and in 2010, he received an early career professional award from Agronomical Society of America, Crop Science Society of America. In 2014, he was recognized by the University of Minnesota as emerging leader in plant sciences. Please join me in welcoming Dr. Jianming Yu.
Thank you very much for the introduction. I appreciate the organizers, and I like to interact with graduate students because I'm happen to be the one that doing similar things like Steve's doing at Iowa State. We like to appreciate your efforts. It's an excellent line of speakers. So today, I have this title, so I have to use the stick again. So it's a pattern discovery, predictive modeling, and then the design in plant breeding and genetics. I have my habit, every time I give presentations, I try to pick terms that I have to study, like pattern discovery or predictive modeling. That happen to be statistic or engineer design, and also the design. So I've given talks where, I talk about the design. Look at this beautiful design, right? Earlier, and all this design, earlier we have two logos of the GWAS peaks, right? Of the CGTA, and then we have the other side of the Symposium series, right? Spade and with plants. So today, I probably will just focus on pattern discovery and the predictive modeling. The reason I put 3 titles here because these are the stable funding, allow us to do this discovery research. Not necessarily the grant proposals, the grant funding. I like to show off this slide. I did not participate in anything. I think Phil or Susan might have reviewed this thing. There's a group of plant science to get together, and then they have this plant science decadal vision, unleashing a decade of innovation in plant science, 2015 and 2025. I strongly encourage graduate students to take a look to see whether you can fit your specific study into this grant vision, right? I'm going to show you chapters here. First one is predicting traits. That's exactly what I'm doing, right? And then the increased ability to predict, look at this, plant traits from plant genome in diverse environment. Wonderful, right? And then the assembling traits. Can we translate assembling traits as plant breeding, right? Symbols together, transgressive segregation, all those finding the mutant alleles might be helpful combined with natural alleles. And then this harnessing plant chemistry, right? I didn't underscore that, but we just find out you can find these new hormones, right? The groups promoting, beautiful. From data to solutions, these days, grad students, you have to struggle with GBS data, genome sequencing, all this RNA-seq, all this long non-coding RNA stuff, right? We don't do that when I was a PhD, right? But I appreciate the grad students' work. And then now here, what we can do is to reimagine graduate training, create this T-training. So we want you to know a lot of things. Whatever we discussed by the speaker, you're supposed to know a little bit, right? And then you go, you're really like a roots deep down, in your specialized area, right? T training. This is a little bit a few years ago. Now you look at this next 10 big ideas for future investment. Obviously, NSF tried to reorganize their grant structure, and we pick 6 research ideas and then 4 process ideas. And I highlight things that understanding the rule of life, predicting phenotype. Again, everybody's trying to predict phenotype, right? And then look at this side, harnessing the data revolution, right? We want you to be good on data because guess what? We're going to have higher capacity to collect the genomic data, phenomic data, and sensor data, and root tracker data, right? Aluminum tolerance data, root length, branch length, lots of data to struggle with. It happened to be what we're doing. So my position at Iowa State is quantitative genetics and maize breeding. I proudly listed the revised version of the position announcement, says to address significant questions in plant breeding. So because sometimes we have so many grad students, we do all sorts of projects, I have to look at this thing all the time. So am I wasting my time doing little things, or am I still carrying some, long-term missions addressing this, major questions plant breeding? Of course, we cannot stay in this old way of variance component analysis in quantitative genetics. We want to combine with genomics data. We want to find the things that we do not already know. I do this because my position has a, distinguished track record. Started with the George Sprague, and then who's really started the corn breeding, right? How to do synthetic populations. And then Bill Russell is the breeder release of B73, which is half acreage of the corn planted, across US. Anna Haller released tons of line, Kendall Lamke. These are the people are telling everyone else how to do plant breeding. We're only listing this maize part. We have another soybean breeder part. Excellent records. I'm here just starting, right? Few years. So, but there's history for me to be proud of. We believe the potential or the power of quantitative genetics, or you can say statistical biology or combination mathematics together to integrate genomics, technology, phenomics, statistics into plant breeding. We really proud of quantitative genetics. Everything actually, in general, everything, this biology getting more quantitative because we have to have the time, labs video, we have to have the growth curves, and then functional principle analysis, right? So our research covered different areas. One is this complex trait dissection. Remember that GWAS plot? We designed this QK mixed model for people to detect the GWAS signals. And then occasionally, we also work on the genes and the genetics to understand the domestication, the traits, the plant height. And also, we try to think about the breeding strategies. When we talk about breeding strategies, we don't have the large resources to actually do it. We just simulate, we try to ask questions. And then we study sometimes genomes and chromosomes, ATGC, the specific compositions, chromosome lengths all together. You can think of, really our study is from DNA, from genes, chromosomes, cells, and then if you'd like to put your favorite species there, it's the same thing, right? So the different scales, right? It's all genetics, right? If you understand the genetics, you can talk to anyone else. It's not like I go to the regular checkup, my daughter, the doctor, the pediatrician saying my daughter's height, right? I already predicted, right? Yes, it's a scientific prediction. So look at it overall, it's just mid-parents prediction, right? You understand mid-parents prediction for your kid's height, right? That's the real geneticists, right? Doctors do not know much better than us. So here, this is what we do, T training, right? In terms of T training, if people in this group work out of the wall, everybody will be millionaires, because in the middle, this gentleman called Harry Stein, so plant breeders can be billionaires, right? He has $3.4 billion of assets. He's a successful breeder in Iowa. He actually opened his facility for us to have the visits. I was shocked. He allowed me to go into his nursery to see his plants. Anyway, that's a raining Saturday morning. I got 6:15. I got a text from my postal saying that Harry's in the field. You can come visit. I was still sleeping. 6:15, Saturday. Anyway, but he's working. So we went there at 9 o'clock, and then he entertained us for 2 hours. That's how we train grad students, to inspire them, and then you can do good discovery in science, you can be billionaires, right? the main topic of my today's talk is pattern. I mentioned the pattern is everywhere. It's a particular way in which something is done, is organized, or happens. It happens in nature, right? The living organism. And if you look at the designs, we have the designs of the different things, right? And then we actually love patterns, but patterns help us memorize, right? We search the patterns. When we see a pattern, we ask why. And then we go deeper for another pattern, right? It's just like on and on the pattern. And then you see, this is the patterns, period, and cycle, right? The patterns can be also summarized in mathematical equations, right? For the primary school kids, they understand the patterns by look at the numbers, right? So what number do you want to fill here? This is probably easy, right? Now, this is also easy. What number do you want to fill here? What number do you want to fill here, right? You can try to get this number. 10 will be here, 4 will be here, and then you have a pattern, right? So humans are actually natural pattern recognizers. From the time we were ancient humans in the cave, we try to understand this earth, right? We try to understand the seasonal changes. We try to find this to feed ourselves, right? To stay away from the animals or monsters we don't understand, right? And then mathematics, physics, chemistry explain patterns in nature at different levels, right? That's why if you're a mathematician, you summarize a pattern in an equation, in an expression. That's just so powerful, right? Patterns in living things are explained by a biological process. We know the central dogma. We know from DNA, RNA, and protein, but there are other new genes. But anyway, there's a general process of the patterns. Expertise, any speakers, any professors, or any soon-to-be PhD graduates, you can be called expert because you know something about patterns in your discipline, right? You know a few things. The advancement is the discovery of new patterns, ask questions behind those patterns. So I dare enough to show some pictures, right? Then Darwin, as this relates to system of culture talk, Darwin discovered the 3 patterns of diversity, right? Species vary globally, species vary locally, species vary by time. That's 3 patterns of the diversity, right? I'm a little bit scared about this mathematician here, right? You can have a formula to describe your pattern, right? Which is E equals mc squared. That's so powerful. We stay away from there, right? Let's look at a very simpler pattern discovered by Greg Mendel. Greg Mendel, I actually teach a class called the Conceptual Foundation of Genetics in Iowa State. We studied Mendel's paper this thick. Anybody want to challenge anything about Mendelian genetics, you better read that paper 10 times, okay? Don't challenge. Tons of experiment was done because there was a paper published in Nature many years ago, the Hot Heads. They tried to challenge Mendel. It turns out to be wrong, right? So he discovered this rules of, laws of genetics. We call laws of genetics, but it's a pattern, right? It's a simple pattern. The first pattern can describe the ratio 3:1. Just pattern, right? You can describe the context. The second pattern is a law of independent assortment. It's a pattern, right? It's a pattern can be summarized as another simple ratio, 9, 3, 3, 1. Beautiful. This is the patterns of DNA, right? Before the DNA double helix was discovered, we first discovered the base complementation by Chagov. The base complementation of that, was incorporated into the double helix model. Okay, a little bit about predictive modeling. This is about a process that utilizes data to train model, forecast the future. You can call this called a magic wand, right? You will just look through everything. Widely used in weather forecast. Right? If I say here, I'm a weather man, right? Try to test this beautiful setting. Insurance, and then believe me that they calculate your risk factors, right? I mean, there's a science behind this insurance, how much they charge you, right? And finance and crime detection. But again, history cannot always predict future. Like what happens, farmers should have been planting in Midwest, but now we have snow still on the ground, right? Weather is hard to predict. But essentially, even though it's difficult to predict, having a biased prediction is much, much better than having no prediction, right? That's the same thing as saying all models are wrong, but some are useful. Because if you have no prediction, you have nothing to rely on. you got nothing to do. If you have just a prediction, even if it's biased, not accurate, the correlation coefficient is 0.2, 3, you have something to rely on, right? I'll shift the gear a little bit about genomic prediction. This is something that we teach and everywhere. This process, basically, I won't go through the details. You're finding the patterns in the genotyping data, in the genetics. You're finding the patterns in the phenotype. Now you're finding the patterns between genotype and phenotype, which allow you to predict, okay? If you trust genetics or trust the genomics, as Susan always say, genomics bring everything to the leveling plan, this equal plan, to do predicting. So here in this genomic prediction, what we do is to a procedure where the genetic merits are predicted for the unphenotyped individuals so that selection can be made. No prediction, no selection, nothing else to do, right? You can have model training process, you can prediction process, you can do selection. So that's a process. So again, I just show these are the patterns behind why genomic prediction works, because things can be summarized into mathematical equations. This equation can have two equivalent models. Here, the left side, the mixed model was set up. The mixed model was set up so that the genetic relationship between individuals are modeled. On the right side, the genetics of the marker relationship was modeled. There's an equivalency between those two models, and there's one is working better than the other one in terms of computational efficiency. My whole purpose is to say that if prediction worked, it's because genetics works, right? We trust the genetics. So here, I'll switch to give a very brief introduction of some earlier work we did. And I have to do this because we're talking about diversity. And I have to remind everyone, this Krasovich is Steve Krasovich, right? This gentleman is supposed to be one of the early pioneers promoting using genetics or genotyping earlier markers to understand the gene bank accessions to benefit the plant breeding process. Of course, later on, this is our speaker earlier. And in this paper, just a seminal. When I was in grad school, in the postdoc, we read this paper 5, 6 times, understand what's going on with the seed bank, molecular marker, unlocking the potentials. This is a science paper, beautiful. And the other PNAS papers, again, recently, just in 2013, this is the championing of the diversity, Susan McCoach. I was promoting that we should be using this natural heritage stored in gene banks. There's about 1,700 gene banks have this germplasm waiting to be utilized. A lot of people don't want to utilize that because it's messy, it's difficult, it's challenging. But everybody was waiting somebody else to figure out a better way to use it. There are ways to use it: integration, conversion, MARS, backcross QTL, integration library. We use coastal mapping. Now we call GWAS. There's allele mining. This represents a continuing struggle, try to figure out better ways to use diversity. They're there. They're just the Mother Nature give us to use for our breeding. If you haven't been a gene bank, this is the gene bank shelves with jars, right? These are the collections from the original places, and then they will stay there. In our frame this small-scale study, we say this gene bank accessions, they have the race classification, collection site, and some information about each of the accessions. And then we have limited capacity to maintain them. We have limited capacity to phenotype them so that we have better information to feed back to the breeders. So when we published the paper, really benefited from Susan's work earlier, the big framework saying we can say, could we use genomic prediction to turbocharge the gene banks? Basically saying that gene bank, they're the materials. Could we actually, we just following whatever Susan is promoting, can we put genomics, right? Genomics allow you to discover the house to be different from indica. Genomics can allow you to sift through all this genetics or the hidden genetic potentials stored in the jars by pumping into more informations in the database, represented by a golden chip here. And here we say, this basically just diagram this whole thing, just saying that if we find the patterns in genomics of these accessions, it can guide us to explore the phenotypes. For individual, we measure their phenotype, we have them. For individual, we don't have the phenotype, because large amount of them. we can predict their phenotype. Again, go back to my statement. Having some biased prediction is better than having no prediction, okay? Okay, this is actually another, the main topic I'll be talking about this here today. I was deciding whether it's time for me to present this work because this is about to be published, but still not published. But because Steve is such a good friend and a student, be wonderful, I said, well, let me just try to present. This is a book. I have a copy called the Triple Helix, written by this gentleman called Richard or Dick Levante. And this scientist is just wonderful. This small booklets, right? How many times I read this one? 1 time, 5 times, 15 times? This book gave me a lot of wisdom. It says that instead of the double helix, right, just DNA or gene-centric, we need to understand the environment, social background, right? The previous speaker touched. It's called the gene, organism, and the environment. All this called a triple helix together, right? I'll actually follow some of our discoveries and follow some of the book, the content of the book. So now we understand the phenotype is between the genotype and the genome or the environment, plus this environmental noisy development process. This sometimes we cannot capture this part. That's give you the diverse phenotypes here together, right? Then I'll start with an example what we did. This is a hidden quiz for grad students only, maybe. And then here is a diagram in this book. This is a classic textbook example showing the genotype by environment interaction or the phenotypic plasticity or reaction norm of the phenotype. This picture was trying to show that it's very challenging to predict the phenotype, okay? Which is true, I agree. Because these are the certain plant species, these are the 7 cuttings. And they planted in lower elevation, medium elevation, high elevation. And then you see the flower and the plant height. This was rank ordered by the lower elevation going down, right? And here is up and down. Here is a down and up. All these patterns that difficult. These are, if you say, if I numerically coding, I basically measure myself off the original publication. And then this is the crazy genotype by environment interaction. So many crossovers. It's hard to predict. Remember the slides? At the end of my talk, I show that there's a pattern behind this whole craziness, okay? Okay, now here is my study. My study, I was at Kansas State as the student introduced me. And then we were in Kansas. We studied 2 specific sorghum variety called the TX430 and the P898012. You see this one flowered early, but this one flowered late. And then in winter nursery, in developing this NAM population, one of the students will be using NAM, and then we found out that P898012 flowered early, and Texas T flowered late. So this is, we talked to breeders, and breeders said, we know that for 20 years, okay. We moved to, I had a career change. I moved to Iowa, which is higher latitude, right? We're expecting to see similar things. But look at what happened in Iowa 2014. And the P flowered early and T flowered late. It switched again. And then we talked to physiologists. They say, okay, there's a summer planting, there's winter planting, there's Puerto Rico, there's Iowa. Of course, you cannot plant in the winter of Iowa. But we can plant summer in Puerto Rico. So Puerto Rico is, subtropical area. We were expecting to see similar things, but here what happened is actually T flowered early again than P. So Puerto Rico summer planting resulted in a switch again. So there's patterns with crazy, right? We understand earlier works by John Mooley's group. They studied the MA1 gene, photoappearance sensitivity gene, from vegetative to flowering time transition. There's another gene also studied. Some of the gene actually homologous in rice and wheat, rhodopsis, many different processes. And then we understand from earlier study, we understand that it's either photoperiod, circadian rhythm, or the temperature altogether to affecting these pathways, right? So here, this is the probably most complicated slides, but because this screen is so big, we can try to understand. These are the planting locations we had, seasons we had. These are the ranked orders of the 7 environments: Puerto Rico 11, 12, PR 14, summer planting S, Kansas and Iowa. These are the geographical latitude, and this circle is representing the day length, right? You see day length increasing. But you see, these are the two parents. Yellow, this blue is T, red is P. These are the 250 reals, recombinant inbred line, the progenies. You see, first time you have a crossover here, right? Second time you have a crossover here, which is pattern, right? It's crazy pattern, G by E. This is typical G by E with switching. Something we can do is sort through these patterns. Something you can do, you can order your x-axis, the same x-axis here, x-axis, based on the average of the whole population, okay? But categorically, you order. Now you can resolve. See that Puerto Rico, Iowa 14, right? Remember that switch in the last one here? You got swapped to the front, stuck together with PR 11 and PR 12. Now Iowa 13, Puerto Rico 14S got switched to this place. You're getting better, right? But then if I replace this categorical x-axis, categorical axis with numerical values of the population mean, here you have this straight line going up, right? Everything seems like it got resolved. It was hidden here. We didn't do anything. We just resorted the data, right? This is also nice and new because in breeding genetics, we call joint regression analysis, just sorting by the environmental means, right? What is new is that our general thinking got changed. So now I switch the gear of the general context of predictive modeling, predictive genomics. Basically, we know the genotype. You can squeeze everything into a one-dimensional axis. You have the environment. There's probably many chromosomes for for the environment, but just let's say we can squeeze them into one dimension. This is going to be the performance, right? This is going to be a performance landscape, kind of crazy, going up and down. And a specific genotype have to pair with a specific environment condition to get this higher yield. It's either here or here. This is the global maximum or the local maximum, right? So basically, if we have our testing site sampled from the environment, have our testing genotype sampled from this axis, we can try to saturate, try to collect the data to map out this landscape, right? So in plant breeding, we have two terms called target the population of genotypes or target the population of the environment, meaning that if you are breeding for certain regions or breeding with certain genotypes, that's your comfortable zone. If you want to go out of the comfortable zone, you may have to explore more genotypes or more environment. So that's 3D relationship. So here, I'll just go through this pattern detection, model fitting, relationship building, predicting, forecasting together, right? So you know that we have limited resources. All we have is tested genotype, tested at the environment. I also tell people that plant breeders or the public research scientists, we hate this G by E because we don't have that much money, resources to testing over like a company, 50 locations, 100 locations. That's what we have here. But to do predictive phenotyping, we need to understand the relationship among this testing the data you have. And then you need to understand how to get testing environment and untested environment connected. Something has to put them connected. Otherwise, you cannot do prediction. And then if we trust genetics, we understand it's easy to collect the genome-wide profile of genotyping data from tested genotype to the untested genotypes, right? So you can do predictions. So what do we actually, in the ag business, what we really want to do is to predicting what are the untested future genotypes with designing crops for the future environment, right? We need to jump from number 1 to number 4. We have to understand the patterns behind all this. Okay, we again go back to our sorghum data. We studied the day length pattern. You see that day length in summer planting in Puerto Rico is actually over the 12 hour, so that's a long day environment. And then it's Kansas and Iowa. You see that in the Puerto Rico in the winter nursery is a short day environment. That's why it gives us this switch of the ranks of the two inbreds and also its progenies. We look at the temperature profile. You can see Iowa 14 is really the cold year. And it's similar to Iowa 13, but then PR 11, 12, other, we try to find patterns here to match our phenotype. Both day length and temperature cannot explain this very well. We also study the temperature cluster analysis. Everything we're doing is just finding the patterns that match, observe the phenotype. Unless you use the performance of the recombinant inbred line populations, we see now Puerto Rico 14S and Iowa 13, PR 12, 14, PR 11, these three together, these two together, these two in the right end, right? We're sorting through this process. So here is probably the most critical slides of the whole talk, is that by combining the statistics and the physiology, what we found out is actually what we call the photothermal time within a period of time. The combination of the temperature and the photo period give us the best prediction. The best prediction can be summarized with this photothermal time for each location. We have 7 locations calculated. These are the population mean of the 7 dots. Look at this correlation. This correlation coefficient equals 0.99, really high. We're basically capturing this environmental variation of this whole population at 7 data points, and then to replace that with photothermal time. Remember earlier slides I have the similar diagram. This x-axis is the population mean, that if you have tested data, you know the average. But now we need to switch to something called environmental index, photothermal time. Now this is a after you obtain this index, it became a performance free. That means you can actually say, just tell me that where I will plant this thing. I can calculate this index. When I calculate index, I can have my prediction, which we'll show that later on. So through the combination of the genomics plus the joint regression analysis, we can combine this into a through this reaction norm parameter estimation process, which I'll explain. This upper left figure, basically, these are now not only locations, they're just photothermal time, x-axis. That's becoming universal x-axis. These are the regression fit value of each genotype. These are basically these 250 lines. Each line actually is a regression fit line described as a reaction norm. You can say this line, which has light blue, is less reactive, less plastic. But this red one, see, it flower early here, it flower late here. It's very plastic. It's responsive to the changing of the environment. And then you can do this from the tested environment to untested environment. You can do this from tested genotype to untested genotype, or you can jump from the tested to the tested untested. You can actually do actual predictions, right? Connect the genotypes, that's genomics prediction. And then environment connection is through this photothermal time. This parameter allows you to put this future environment or tested environment together to model. You can also try to do this prediction. Remember my slides on the left mixed model, right mixed model? One was written on the individual, one was written on the genome marker, same thing here. You can do this joint genomic regression analysis from genome the marker effect, right? I'll show you this one. On the upper left, remember earlier figure I had, I'll remind you again. Here, each line represented an individual, a genotype. Now here, each line representing a marker, a GBS marker, a SNP marker. This marker tends to have different effect depending on the environment. Those genes have different effect based on the input of the environment. These are genome marker index. So basically, if you have this already done, then you give me a genotype, you give me a new individual, I can look through this marker profile and then add all this together at each specific input of the environment. If the environment changed, I go here, I look at this genotype profile, I summarize this all together here. So you can predict from a relationship of individuals or from a marker profile. And then these are the same settings. And then we see that the overall prediction accuracy, even the most challenging case is 0.86. But within each single environment, the prediction is only relevant locally. It's slightly lower than the overall prediction of everything together. The reason why that happens. Okay. So, okay, we said history cannot always predict the future, right? Let's look at the future. So we actually have our model developed with the first 7 environment. We made the prediction. Remember here, all these shaded areas are the environment we detect this, we establish the model. And then Iowa 15, Iowa 16 are the empirical validations that we grew out the same population, we try to validate our prediction. How do we do that? Because we have all this profile, we just calculate the photothermal time of the Iowa 15. Look, Iowa is very greatly, right? We just go up, that's our prediction. And then here, we go up, our prediction. What we observed, so this is the predicted of Iowa 15. This is observed. We're a little bit tilted. We predicted a little bit early, but then flowered a little bit late. Look at Iowa 16, everything was right along the diagonal line. So these are the predicted flowering time expressed at the green degree days. So if we predicted right on target, that's around this diagonal line. If a little bit off target, that's your, deviate one from this diagonal line, right? Again, sometimes we do predictions. The prediction can be scary to some biologists because they say, well, if you do predict, what else we do? I said, no, you have a lot of things to do, right? So this is true because I hear a lot of negative comments about prediction. But look at that ASPB, that decadal plan. It's everybody, you do predictions too, right? Everybody do predictions. So you look at this QTL mapping result. You have the MA6, MA1, FL8.1. This is the ELF3 gene, flowering time gene. The reason we can put exactly this gene because many of this, a number of this MA1, MA6, and FT, these genes has been identified. Somebody else cloned this gene. So all we need to do is to sequencing through and identify the functional polymorphisms. So I can see that these genes are functioning. It is genetics interacting with the environmental parameters, giving me this prediction phenomena. It's not just a prediction, non-parametric or something you cannot understand. We did all this to prove that everything is working beautifully. Now here you can see that specifically 5 locus. Let's say look at MA1. MA1 is a major effect gene, photoappearance sensitivity gene. In the tropical environment of some of the numbers, number 1 and 2, Puerto Rico 12 and 11, 12, is actually have a negative flowering time. It promotes flower. But in this Kansas environment, it actually increases flower. So this is something that's very difficult to actually, the genetic effect changes depending on the environment, right? You see that some of them you can detect in some environment, but not detect in other environment. Some of them, it's a conditional neutral. That means you detect in other environment, but not in this environment. Again, like what our previous speaker detected, the epistasis. If you actually do a 2D dimensional scan of the 10 chromosomes, we find this chromosome 6, MA1, and chromosome 10, FT gene, consistently showing an epistatic interaction, even if you analyze all together or some single environment. What is amazing that sometimes this gene, the marginal additive effect cannot be detected, but epistasis is still being detected. So that means the whole network is actually functioning. These are the specific genes. We validated these specific functional polymorphisms. These are this is actually homologous to the rice flowering time gene. So this just show that some of the things that grad student has to do, right? You do this large scale sequence alignment to detect this insertion, stop codon, and then to, there's an intron here, insertion here, there's a try to find the functional polymorphism of this FT gene, ALF3 gene. We got all kinds of things to prove, geoimage to prove, because we want to connect the genotype by environment interaction into the gene by environment interaction, right? Genotype is genotype, the whole thing. But gene, that's the specific polymorphisms here. Again, we said you can change the phenotype across environment as a reaction norm. Remember that reaction norm, flat or responsive? These reaction norm parameters have two parameters. One parameter is called intercept. One parameter is called slope. If you map those intercept slope with the mapping with the intercept slope, you do QTL mapping, you find the same set of genes, right? You do this inclusive composite interval mapping, you find the same set of genes. Again, this gene by gene interaction here also affect intercept and slope. So we just prove that there's a genetics behind whatever we observed or whatever we designed to make a prediction. Here, the more research we do, we say we better check literature, right? Because we feel as we relate this specific environmental parameters to the observed phenotypes, we better read some of the earlier study. It turns out that historians, they study genetics too. This paper said R.A. Fisher and Lansard Hodgkin, origin of genotype by environment interaction. It turns out that Fisher actually, in his nonlinear interaction of heredity and environment, he did not like this interaction because he wanted to partition the variance in different components, right? If you have interaction, he wanted to do a transformation to minimize interaction, which is okay. And then this another expert called Lansard Hodgkin, he's a developmental biologist, but he's also a statistician. He focused on this interdependency between the nature and nurture. He wanted to stress the developmental noise. So there were two terms called biometrical concept of G by E, biometrical B. So, and then developmental concept of G by E and over D, developmental. So we saw there are other examples previously, but our example says that it is a developmental concept of G by E eventually give you the final observation of the biometrical concept of G by E, sort of unify this concept together in our simple example. So I showed that you can do a integrated analysis to study the gene and environment together. That's what we were inspired by that book, The Triple Helix. Because we can look at the known or candidate genes, we can study this genotype by environment interaction at the genotype level, but also at the gene level, and plus the G by G interaction. So here's a diagram understanding what we think is through this nature-nurture debate, 1910s and 1990s, and by genotype by environment interaction from 60s to 2010s. We started to do a lot of genetics, but we do not actually incorporate whatever we did with this environmental factors. We do Goose chamber with fixed settings, but then now when we're doing this not only in the individual genes, but gene networks, plus we can quantify this environmental input together. Quantification is critical because otherwise it's floating around. And also because the environment is time series, right? The daily temperature changing through the season. The photo period also changing through the season. You have to quantify this all together. We think we will be able to help us understand this G by E, this crazy crossovers we see. This is another review was done earlier by Thomas Younger's group. And here they showed that this is just an investigative review. In some environment, you can have this called antagonistic pleiotropy. In some genes will show this conditional neutral. Yes here, not detected. And here, both detected, but differential sensitivity of the gene, so that QTL is come and QTL is gone, right? And sometimes you have no G by E because QTL is either consistently detected or none of them detected, right? So that's the conceptual work. But if you look at what we did is actually, if we can quantify the environment into the a series, you can see that sometime of this, these are the conditional neutral because not detected here, detected here. This is a mechanistic pleiotropy, negative, significant, positive, significant, right? So all these things actually is together. It's just Mother Nature interacting with this plants all together. It just depending when the plant, again, from a quote, plant are mathematicians. They know how to do time average. They know how to do differentiations, also to do integrations. Plant just react all this, we call plant is a perceptron, react to this environment, deciding it's, whether it's flower early or flower late. Amazing. So these are the specific 5 genes. You can see that their QTL effect can be, this can be depicted a different way with this. And if you want to look at the genome-wide markers, the genome-wide markers, this effect can be positive or negative depending on specific environment. They can be all put together. Again, we call this called SS, IS, OT, rigid SS, because these terms are actually not the plant genetics terms. These are the production agriculture terms. So site specific, right? I want to do a prediction for that site because the site, with the site, I know the historical average of the temperature, photoperiod, soil type, everything. And then if I know the site, I can do predictions, in-season prediction. If my season started, I would stop using the 15-year average. I would switch off to the new season, early season data to update my prediction like Dan showed earlier, right? In-season prediction. I can adjust my prediction in a local region because of the weather pattern. On target is whatever we want. This is a local environment, it's global environment. We want to exactly predict the performance, not the ranks only. And then this will be off target, that will be off target, right? And then this whole genome, again, we show this again. If you use the whole genome prediction, that's a whole genome prediction. If you only use QTLs, because this data can be used both for QTLs and whole genome. QTLs are the specific combinations. You get lower predictions. If you use whole genome, it's much higher predictions. And simple, straightforward is because our interaction with crop modelers, they inspire us to look into the quantification of this environment together. We think in the future, everything has to go through the crop modeling, but does not prevent us from, right now, to use a simple, straightforward method to simplify the crop model to capture that. So again, so this relates to design thinking, data mining, right? Thinking about our future environment might be on this axis, right? These are the future genotypes. How do we strategically design our experiment? Are you testing the materials at the best testing site? Are you testing the site at the testing site? Are you bringing out the genotypes that will give you maximum information, right? It's an optimization of the genotype to be tested and the testing site to be tested. You map out the whole diagram. I'll throw one more kind of crazy ideas. Winter nursery data can be used for prediction. Oh, that's not true, right? We go there just to get the more generations. If we figure out the patterns behind this change of the reactions, we can leverage partially of the winter nursery data to do prediction. Okay, now it's one of the last few slides, okay? I see how much I love this book. I showed that my respect to the Dick Leventin. I read this book many times. I'll be reading this again on my way back to Ames, Iowa, right? Just so much information there. I show this figure again. Now, I have to criticize a little bit, right? I feel bad I have to do this. So here, this is a picture to show that G by E plasticity is difficult to predict. You stare at this picture. You stare at this picture again and again, right? You look at this number 2, number 3. Are these two roughly similar? Yes? 2 and 3. They have a similar response, right? You can do the same thing for other ones, right? Let's say you look at the 5 and 7. 5, 7, you look at this, right? What about 1? What about 1, 5, 7? this seems like follow similar patterns, right? We can dissect this whole thing into different subgroups. It's like doing a cluster or doing a principal component analysis or doing a structure analysis, let's do that. So here, I already reshuffled everything because I shuffled lower elevation on the top, high elevation in the middle, medium elevation in the bottom, right? I also shuffled the columns. I'm photoshopping this whole thing, right? I'm allowed to do that because I have to show the pattern to you, right? Now you look at it closely, 157. They're similar response. And then if I showing that numerically, they're going down, a little bit, right? They're similar patterns. 2, 3, very similar, right? If I do 2, 3 together, look, they're not crazy, right? 4, 6, So here's my slide. There's patterns. Behind this diagram was developed to show us there's no pattern, right? It was crazy up and down. That is difficult to make a prediction. But if we study hard enough, we find the patterns, right? That's exactly the pattern that works in my sorghum prediction, right? My genotype was responding slope and intercept. I can dissect them into, and there's genetics of those QTLs that function, those genes functioning behind my pattern. And there's also, my pattern was developed because of the genotype by environment interaction altogether, right? So we got funded by a lot of agencies. I showed the main work was done by Xianran and Ting, and then also Xing Li. And over the years, we talked with the crop modelers. We talked with quantitative geneticists. We talked with biologists. And then Pat Schnobel is the director of the PSI, funding long-term research for us to do predictive genomics. We're very excited to do this type of research. We also get pioneer funding and collaborative research with Elliot Hefner and Tabari. We appreciate everyone. My slides most of the time kind of crazy and messy. I have to entertain my audience, right? Let's entertain you with the last animation. Not as good as Theo Benth's the movie, but we decided to do something, right? Live validation performance forecasting. Here we go. This x-axis is predicted flowering time, y-axis is observed flowering time. If I'm seeing any dot, I bingo, right? Bingo once, bingo twice. I keep waiting, right? Because my prediction was done before the season. I'm just waiting for them to flower. Please flower along slide. Bingo here again, right? Look, right? Look. Not too bad. This is annoying, right? There must be some genetics and patterns that give me that annoying dot. Okay, that's all my presentation. Thank you.
So when you saw this clear pattern with flowering time for these different environments, did you also go back and do you also do an end of season phenotyping for grain yield and stuff like that? And did you see any pattern on that, or it was just on the flowering time?
Challenging questions. We try to stay away from green yield, right? We try to do the number one trait is the flowering time. Flowering time, for people doing flowering time, they say flowering time decides everything. Right? I mean, we show that flowering time decides some of the root too, right? Root biomass partitioning, and then, over the winter too, right? No, we did not do any green yield because we think strategically, I refuse to do that because it's challenging for me to see the patterns. It's relatively easier for me to see the pattern in flowering time because here, we construct the photothermal time environment index only takes the temperature and day length, right? If the yield come into play, the moisture, the nitrogen, I will not get these beautiful patterns to get everybody happy first. But we believe everything we did can be modeled, can be expanded to much larger traits, which we did. We have to show in other data sets that green yield, you can have a x-axis constructed. You can have this overall geographic predictions to a certain degree. But the prediction accuracy is not as high as flowering time. It might not be 0.8, it might be 0.6 or 0.5. So we can make some progress over there. That's an excellent question. Thank you.
Any other questions?
Emily? Yes, that's a perfect question. This is so right now, we model the environmental mean of the 7 locations, and we get a very accurate estimate. We suspect that light intensity might be the ones that change our prediction, lower our prediction in a specific environment, like the particle 11, 12. There, if we off the target a little bit, that means there's, okay, if our predictions stay on this line, that means our environmental index constructed perfectly matching. But if we off target, meaning that there's spatial environment factors come into play that tilted, move this away from the prediction. But in the future, we'd love to do that. Excellent question. Please.
I'm not sure if I missed some information, but it seems like all the experiments are operated in the field, right?
Yes.
So have you ever tried to test like the greenhouse test and to see like if you somehow make some factors different and in the greenhouse, if this predictive field?
Whatever I showed today does not exist in textbooks or publications. It's brand new discovery. We believe that if people doing this specific greenhouse studies or Grist chamber study, circadian rhythm, gene expression, if they take this one, they can design excellent research, studies to mimic what we did, even much better. However, what we also did is we went to the literature. We summarized about 40 different experiments that people have done for flowering time in different species across 20 species. There, the temperature setting and day length settings are fixed, right? They're not a specific growth region or growth period. We do the calculation. Many species follow the photothermal time. Some species follow the temperature only, but not photosensitive. And then this result was actually was showing as a supplementary table in our manuscript. Yeah, which we already proved with existing data, which we can be used to do more research. Excellent question. Thank you. Yes. Well, I spent 5 hours to make sure I understand what's going on there before I, came here. Oh, sorry. How do I get to the? I think the question was actually one of the last prediction. We had a problem of that tilting. Should we go there or should we? What do we do actually? Can we quickly go to the? Let's see. Well, you get a review of whatever I showed, right? Like I get a second chance to present, right? Yeah. But that was an excellent question, I have to say, because 15 bothers me that we were promoting our predictions on target, we were off target, right? This is what you're talking about, right? This is because when we try to identify the window to summarize photothermal times, it's a 20-day window. It turns out in Iowa 15, the weather pattern changed. We have a low temperature and go up, low temperature, and then go up. Well, the way we develop this model, we average across whole season. But in Iowa 15, it's actually the temperature climbed up. So it actually, the flowering time should be late. Because the way we did that, we predict to be early. That gives the tilting. We understand why it happens. Again, that's why my other statement. We have a pattern, we have a rule, we find exception. Then we refine our pattern description to cover that exception. It's an ongoing process. Excellent.
Thank you, Dr. Yu.
Okay.
So thank you all the speakers for the session. So we will adjourn and we have poster session for an hour. We'll reconvene here at 3:45. We have snacks in the glass box outside by the atrium. So please join me in thanking Dr. Yu and thank you all for coming.